Next questions and answers. Globular and fibrous proteins related. So hemoglobin is a D. That means Q, R and S are right. Q, R, S, right. So an oligomeric protein, a chromoprotein and an allosteric protein. Both are correct. But it's not a monomeric protein. And that's wrong. All other options are correct. Oligomeric protein, a chromoprotein and allosteric protein. And the quaternary structure of human hemoglobin is best described as a The quaternary structure of human hemoglobin is best described as a tetramer of two different subunits. Yeah, that's right. When GLU at the sixth position in the beta chain of human hemoglobin is replaced by valine, that will see. That means quaternary structure is significantly altered. Then the difference between the R and T states of hemoglobin is P. Oxy and deoxy states. And binding of oxygen to hemoglobin occurs when Fe is in the following oxidation state. Which oxidation state? B is the correct answer. B plus 2 oxidation state. The advantage of hemoglobin having a high histidine content. The advantage of hemoglobin having a high histidine content is 6C. Histidine imparts buffering capacity to hemoglobin. Histidine imparts buffering capacity to hemoglobin. Then which one of the following statement is false? Actually, the D means none of the above is false. That means everything is correct. So homozygotes for HBS results in an elevated BPG concentration in their erythrocytes. So homozygotes for HBS results in an elevated BPG concentration in their erythrocytes. Hill constant N can never be larger than the number of ligand binding sites on the protein. Peptide groups, with few exceptions, assume the trans configuration. Then none of the above is the wrong answer. Then which of the following statement is or are true? There can be multiple statements to be turned out to be true or false. Okay. So C is the correct answer. P and S are true. So P means cooperativity coefficient is a parameter in Hill's plot. Yeah, that's right. Then yes, S means at low pH oxygen binds to hemoglobin weaker than at high pH. Low pH in the sense acidic condition. Oxygen binds to hemoglobin weaker than at high pH. And which of the following statements are true about myoglobin? Actually, the third one. That means C is correct. P and R are correct. So P, it is a monomeric protein. Which one? Myoglobin. Monomeric protein. And R in the sense the molecule has 8 major helical segments. Then the residue histidine can act as a ligand for the heme ion in proteins such as myoglobin, because what is the reason? 10b. One of the nitrogens has a lone pair of electrons available for coordination. Then which one of the following statement is incorrect? So we will be excluding that. 11. C is the incorrect one. So all others are turned out to be correct. MB consists of a single polypeptide chain in addition to a heme prosthetic group. The red color associated with the oxygenated forms of MB and HB is due to heme. And the alpha and beta chains of the HB tetramer are slightly shorter than the single chain of MB. But HB has a higher affinity for oxygen compared to MB as seen from the oxygen binding curves. That's not correct. Okay. Which of the following statements about oxygen binding and release by hemoglobin is correct? 12. Correct answer is B. B. Lowering the pH. That means 12. Which of the following statements about oxygen binding and release by hemoglobin is correct? Which one is correct? 12. B is correct. Lowering the pH accelerates the release of oxygen from oxyhemoglobin. Lowering the pH accelerates the release of oxygen from oxyhemoglobin. Then the hemoglobin consists of 2 alpha and 2 beta chains, each of which has a heme group with an iron atom at the center. The iron in the heme binds to an O2 molecule. The hemoglobin consists of 2 alpha and 2 beta chains, each of which has a heme group with an iron atom at the center. The iron in the heme binds one O2 molecule. Therefore, one molecule of hemoglobin can bind 4 oxygen molecules. Indicate which one of the following statement is not correct. 13 D is not correct. That means most of the CO2 produced by the cells are transported in the blood in form of bicarbonate ions. That is not true. But all other statements are turned out to be true. So changes in the pH of blood affects the binding of oxygen by hemoglobin. Yeah, that's right. Carbon dioxide from respiring cells diffuse into the blood and lower the pH of the blood, which enables the hemoglobin to release O2. Then the CO2 can bind to the iron of the heme, which is free. Then in sickle cell anemia, the basis of the malfunction of the hemoglobin molecule is 14. Correct answer is turned out to be A. So, substitution of a single amino acid. That is the malfunction. Then which one of the following describes a major effect of sickle cell anemia? 15C. Decreased solubility of the deoxy form of hemoglobin. Decreased solubility of the deoxy form of hemoglobin. And which one of the following pairs refers to paralogous proteins? So the hemoglobin consists of 2 alpha and 2 beta chains, each of which has a heme group with an iron atom at their center. And the iron in the heme binds one O2 molecule. Therefore, we just mentioned this. Then, which one of the following pairs refer to paralogous proteins? 16. Correct answer is A. Rat myoglobin and rat hemoglobin. Myo and hemoglobin. Paralogous proteins. An example of a structural protein which is also an enzyme is 17. Correct answer is found to be B. B means myosin. Myosin is both an enzyme and also a structural protein. Structural protein and enzyme, myosin. Then most abundant protein in the human body is 18C. See, collagen. Collagen. Okay, collagen. Then an example of a trimeric protein is 19D. D, collagen. That's a trimeric protein. Then collagen is best described as an alpha helical structural protein or a coiled coil found in hair, a cross-linked globular protein, or a triple helical fibrous protein. The actual answer is D. That means a triple helical fibrous protein. Then next 21st question. Collagen is a fibrous protein consisting of 3 peptide chains. Collagen is a fibrous protein consisting of 3 peptide chains intertwined in a triplet helix. Which repetitive motif is responsible for collagen's ability to form the triple helix? 21. Correct answer is actually C. C in the sense glycine and XY. Which one of the following is unlikely to be water soluble protein? Which is unlikely to be? 22. C. Collagen is unlikely to be a water soluble protein. So which one of the following is not correct? So that means other proteins are water soluble. Which one? Ribonuclease, myoglobin and lysozyme. Then which one of the following is not correct about the collagen triple helix? 23D. 23D is a alpha helix. Yeah, that's a wrong answer. That means all other statements are correct. It's rich in glycine, is rich in proline, is rich in hydroxyproline. But it's not an alpha helix. Then choose the incorrect statement with regard to collagen molecule. Incorrect statement. 24C. It contains less hydroxyproline than elastin. Then it is formed from 3 alpha chains. Glycine accounts for 1/3 of amino acids found within collagens. Absorbance is essential for hydroxylation of protein. Which one of the following statements is true about the structure of collagen? 25A. Fundamental unit of collagen is Tropocollagen. Yeah, that's right. All others are turned out to be, yeah, that's not correct. P, Q and R are correct. Fundamental unit of collagen is tropocollagen. H bond between 3 alpha chains hold the helix together. P, Q and R. R is also correct. Rich in proline and glycine. And which one of the following cofactors derived from a vitamin is involved in the formation of hydroxychloroquine during collagen synthesis? 26. Correct answer is D. That means ascorbic acid. When collagen occurs in different forms, which is usually classified on the basis of the 27th D types of peptide chains present, then choose the mispair. 28 B. B means collagen. That's not matched. Okay. And which one of the following is not a feature of alpha keratin? 29 D. D means presence of intra-chain SS crosslink. Presence of intra-chain SS crosslink. That's not a feature of alpha keratin. Then desmosine. Desmosine is 30. Correct answer is B. B means P and S. P derivative of 4 lysine residues. S means is found in fibrous protein elastin. Then disulfide bonds do not form in the cytosol, but keratin intermediate filaments in the skin are cross-linked by disulfide bonds. It can be explained by the following. 31 D. Disulfide bonds in keratin are formed after cells have died and therefore cannot maintain the reducing environment in the absence of metabolic activity, and cellular contents are oxidized. The next, protein techniques, that means electrophoresis, chromatography, spectroscopy and configuration. So first question is related to protein techniques. That means the isoelectric point of alanine is 6. If alanine is dissolved in the buffer of pH 3 and subjected to electrophoresis, what will happen? Most probably it will not migrate to either anode or cathode. Okay. Then the rate of migration of a protein in STS polyacrylamide gel is not influenced by 2C, pore size of the gel. Oh, that's a new information. But size of the protein, charge of the protein, strength of the electric field, that all other factors are influencing. Then which one of the following statement is false? 3D is false. Bromophenol blue used as tracking dye during electrophoresis. No, that's not correct. So beta mercaptoethanol acts as a reducing agent and used to break disulfide bonds. Chemasi brilliant blue used for staining proteins. Yeah, that's right. Sodium dodecyl sulfate is cationic detergent. Yeah, that's right. And then STS is used in polyacrylamide gel electrophoresis of a mixture of proteins for their efficient separation on the gel. SDS in this experiment is used to for B means stabilize the protein. Okay. And sodium dodecyl sulfate upon adding into water can form spherical micelles. In this micelles, the hydrocarbon tail would be 5. Correct answer is A. A means cleaved on micelle formation. Then if the pH becomes lower than the isoelectric point of a protein, then how will the protein respond in an electrophoretic system? It will wait a minute. Yeah. A. Correct answer is A. Become denatured. So which one of the following electrophoretic procedures does not depend on the charge of the protein? 7. Correct answer is actually C. That means polyacrylamide gel electrophoresis and sodium dodecyl sulfate. That doesn't involve the procedure. Electrophoretic procedure does not depend on the charge of the protein, but all others are important. So moving boundary electrophoresis, zone electrophoresis, isoelectric focusing, everything require charge of the protein. But polyacrylamide gel electrophoresis in sodium dodecyl sulfate doesn't involve charge of the protein. Then in two-dimensional electrophoresis, proteins are separated 8. Correct answer is B. B means first by their mass and then by their charge. Then two-dimensional 2D gel electrophoresis performed under denaturing conditions can be used to separate proteins according to each of the following characters. Which one? 9. Correct answer is A. Subunit molecular weight, density. Subunit molecular weight and density. And a mixture of amino acids consisting of glycine, lysine, arginine, histidine, aspartic acid, and glutamic acid was placed in the center of a paper strip, moistened with buffer of pH 6 and electric current applied. The migration of amino acids was as follows, like this. "16. Correct answer. It's not available. I'm sorry, it's not 16. It's 10. 10. Correct answer is B. B in the sense. Aspartic acid and glutamic acid remained near the origin, and lysine, histidine, and glycine moved to the anode, while arginine moved towards the cathode. The next 11th question. SDS and NaCl are used in the purification of genomic DNA from peripheral blood samples. Their function in this process is respectively 11B. B in the sense salting out of the protein, denaturation of DNA. The relative viscosity of a protein solution is affected by 12D. D means temperature and shape.